up. So now we've seen a few different varieties of brittle stars or ophiroids.
They often have either a polychaete or another crawler on them.
Okay, time to go, guys.
Yeah, Thank you, Dave. Yeah, that's great. Yeah, there's
An
a lot of
isopod or
animals associated, and whether they're being consumed or future prey for the sponge, or if they're basically using the sponge and actually eating particles or anything that's trapped on the sponge off as food remains to be seen. But yeah, they're a type of demosponge, different group than the glass sponges, as I mentioned, and the type of carnivorous sponge, I think it might be
I keep doing this as you
a
come up and down.
cladorhizid.
I'm about 60 down now, and I'm
It's more
tilting up.
candelabra shaped.
That's about 45.
Oh,
Lisa
great.
says it has an
I'm gonna do a round
isopod,
of,
ophiroid,
checks,
and
so
a polychaete on it.
might be distracted, okay?
Thanks, Lisa.
Roger.
Yeah, there's kind of a candelabra-shaped cladorhizid sponge that has been imaged from the abyssal plain in the Clarion-Clipperton zone, so that's my best guess for that one.
Steve from our science portal is saying that it looks close to the asbestopluma, but very lyrate and branching pattern.
I'm going to take
Thanks,
camera
Steve.
up here. 6. No, but you can see that the gauge isn't like 0. You know?
And Asako confirmed that this is the first time we've seen
You can
it.
see if the needle were in the 0
That's good.
area.
I haven't been missing anything while I've been sleeping. That's good to know.
It's coming out just for our shift.
There's another starfish.
Yeah, looks like a Brisingid sea star. Its arms waving in the currents. Flyby is good. Thanks. Thanks, Dave.
Okay, I have a question for someone in the front. What is life like on the ship? Are there any tricks you learned from your time on the sea that maybe you didn't know when you first set sail?
Cannot confirm nor deny anything.
No tricks.
Josh? Anyone?
I'll say a lot of people don't realize that if you wedge a pillow under your mattress when you're rocking in heavy seas, it creates a little pocket. And then you don't get thrown around all night.
Nice.
Like a little cradle.
Bring your own candy because it will not be provided.
True.
Chocolate.
I learned the hard way.
There is no chocolate on this boat unless you bring it yourself.
Bring your own chocolate.
You should keep at least one comfort food. It's funny because everyone has their own little comfort. You know, like I often see someone come out and they have like their own coffee grinder and their special, you know, like high premium coffee. So
guilty here.
Yeah, you know, so everyone has their own little thing, and it's just a matter of getting something that kind of reminds you of home and keeps your mind focused.
I don't drink coffee, so I lay in a supply of Diet Coke.
It's some Diet Coke.
Yep.
Oh, now I know who to hit up for Diet Coke.
Yeah.
It's running low. I'm going to have to switch.
I know, yeah.
I have backup sugar Coke that that I have to switch to.
Man.
Backup sugar Coke.
Yep. I bring my own pillow, my own sheets.
Oop, your own sheets is a good one.
Yep.
Yeah,
Yeah.
creature comforts.
I like high thread count sheets and pillowcase. Actually, I have a pillowcase that my wife made for me that's
What's
special.
That's
that
nice.
dark, straight thing to the left there?
I will say there's different sized beds on the Nautilus, so I'm...
True. Tricky with the sheets.
Yeah, you just turn down the king size bed.
Yeah, I never get the king size bed. That's
What are we looking at here?
VIPs get that stuff. Not
It's like
me.
a large...
I don't know, we've been seeing
think
large
it's
shark's teeth
Is there a
throughout the
shark's
dive.
tooth?
Yeah,
Maybe.
so it might be a shark's tooth.
Alright, we've got time. Let's have a zoom and see what we got.
You're
Yep.
a VIP to me.
Give me a sec.
Yeah, you're a VIP to us, Dave.
Well,
Yeah,
thank you.
shark's tooth.
Thank you, I
Yeah,
appreciate
it
it.
looks like a shark's tooth, folks. Well coated in
All right, I'm back with you, Dave.
manganese,
Okay, mate.
iron oxide.
Alright, on the
Forward.
move.
Thanks. Thanks, folks.
Camera's still at about 45.
Cool.
Seems like the nodules are all really similar sizes and shapes. Yeah.
It has been consistent since we landed. Just a very high coverage with all of them being fairly spherical in shape.
Yeah, we have to work on the descriptors for those train conductors that are listening.
You notice I was trying to add that in there, specifically for the train conductor.
Exactly. Now that's a good tip with the pillow underneath the mattress for it not to rock as much.
Yeah,
Get some leverage in there.
it works really good. And then like under your actual pillow too, that works, yeah. Especially when we start getting thrown around and like you find yourself sliding.
Right. Actually, yeah, one of the people in the VIP rooms with like the larger beds was rolling around too much, so she ended up moving her bed onto the ground.
Oh, yeah.
VIP problems.
VIP problem.
You didn't know the pillow trick.
Yep, she needed a smaller space to roll around in. The bed was was off the wall, so she needed more barricade.
Yeah, definitely bring snacks. What else? Some people started their...
Another
Another
sponge, I think,
yeah.
on the right.
Some people started waking up for their shift times before they had their shifts, so they could get on schedule.
Oh, yeah. That's another good thing to get used to.
Are we seeing some holes and burrows right here, or is this just the way the shadows are hitting the rocks?
It's a good question. I haven't seen any burrows specifically, but it looks like the nodules are kind of piled up
Yeah.
in some places, and John and I were talking about that. I'm not sure if that's due to animals moving underneath, like over time that those infauna could be moving things around and through their bioturbation and cycling of the sediments, or some other process that's happening, because the nodules are loose, they're not cemented in place here. So if we were to collect, you know, with another vehicle, we could just kind of pluck these off the seafloor. And in order for them, I guess, not to get cemented in place, there has to be some level of movement. Much like we have to stand up and move every once in a while, or else we feel like we're cemented in our chairs. But what's creating those piles of nodules is a bit of a mystery. A lot of speculation going around, at least by me.
Lisa said, I bet the shrimp practice their acrobatic acts by swimming through that. And then we have a question about the current from Cape Town, South Africa. You mentioned a current on the seabed, and how fast is it? And does the ROV create a current as it passes?
At the moment, there basically is no current. If there is, it's very, very, very slight, to the point where I can't feel it on the vehicle at all. ROV will create a little bit of a wash just because you've got the thrusters running and just hydrodynamic flow as you're pushing through the water, but not a lot. Occasionally in the stern camera of the ROV you'll see some sediments kick up. In fact, you can see it right now. That's just from us moving really close to the seafloor. But it's pretty gentle, quite benign to be honest at the moment.
Look at this sponge coming up.
Yeah, we've been seeing a number of those sponges throughout the dive, and that looks like another, maybe a dead sponge coming up dead center.
You wanting to have a look or fly by?
No, we can fly by. Thanks, Dave.
Sweet.
Looks like the remnants of a glass sponge.
I agree.
Cape Town, South Africa. It must be related to John's wife.
Nice place. Been there a few times. In fact, kicked off on the first endurance expedition out of Cape Town.
Oh.
On the Agulhas 2, their national icebreaker. Some ship that is.
I'm amazed by the agility of that ship in the ice.
Incredible, mate.
One of the things that really impressed me though is when they get stuck a little bit, they pick up a big fuel tank on the massive crane arm and heave it back and forth just to rock the ship.
Rock the ship a little, yeah.
Yeah.
I've got video of us doing that. We got stuck for a couple of days when we were down there.
Yeah.
Well, that must be exciting to see.
Yeah.
Waving back and forth.
Big 40-ton.
Yeah.
It's a
drum of
massive
fuel.
arm.
Wow.
Sling it in a crane and just throw it around the bow of the ship.
All right, we have actually a really good question. Might there be any animals living underneath the nodules?
For sure.
For sure, yeah. There's lots of animals that we can't see.
Thought I'd turned SPL off.
Yeah, I agree. I agree.
Talking to myself.
That's my my go-to phrase. Yeah, the communities of organisms that live on the nodules themselves are called epifauna, and then those living in the sediments below the nodules are infauna. Essentially, our cameras aren't super high focus and high resolution to be able to go in and dig into those sediments here, but there's a whole community of organisms from different types of polychaetes and crustaceans and other types of worms that live in the sediments that provide a variety of different functions and roles in processing organic matter and serving as food for other organisms. They're part of the ecosystem, and we use a variety of different devices, including ship-deployed box cores and push cores from the ROVs that we've used in the past to collect those samples to better understand those organisms, the community, and they're part of the diversity here. That's a group of organisms that my lab specializes in. We've done work on infauna and all the main major ocean basins except for the Indian Ocean, and been really interested in characterizing those communities across the globe. They provide indicators for different ecosystem changes. So they're useful in terms of understanding ecosystem health. Something's swimming around, looks like. I don't
Some black snow.
know. Maybe we're seeing shadows or something.
Okay, so those are primarily studied using the samples then, rather than the imagery.
Correct. When we've been zooming in on the nodules themselves, we've been able to see some types of what I'll call macrofauna that are epifauna. So they're living on the surface of the nodules, epi. But for us to better understand the infaunal communities within the sediments underneath the nodules, we would have to collect. Yep. But these images provide us a chance to better prepare and plan for those kinds of collection missions in the future.
And then what would that look like once you've collected a sample?
Okay, so once we collect the samples, if they're push cores are basically tubes of mud that are brought back on deck into the lab on the ship, and then looks like another stock sponge on the left, and then we essentially use a device called an extruder. It pushes the mud up from the bottom of the tube to the top, sort of like a push pop, like you would eat from the ice cream store. And then we slice the mud at different depth intervals so we can understand what's living at the sediment surface and then deeper depths within the sediment column. We also process the sediments for chemical analysis to better understand the environment that's associated with those infaunal communities. And then the sediments are preserved in a way for us to then sieve them and then pick the animals underneath the microscope. In addition to that, we've subsampled sediments to do genetic analysis so we can then further ID them. So it's a process, but we have a whole group of scientists back on shore who help us process these samples and then identify the animals.
That sounds fun.
A lot of microscope work.
As a little kid, I would collect things from my yard and look at them under a microscope. Usually they ended up being trash.
Oh.
Because I was attracted to the more like glowy bright
things.
colors. But
Everything's shiny, even if it's just a plastic wrapper.
I would have loved to look at some of this stuff.
Yeah, I agree. Someday.
Someday. That little girl.
I'll have to get started on that underwater microscope for you.
Yeah, thanks, Aaron.
Thank you.
There are cameras that have been developed that basically can be depressed into the sediment, and then you get a cross-sectional view of the sediment itself. It kind of creates a slice into the mud. You shove it in, and then you can see that bioturbation that I mentioned. The animals themselves create structures in the sediment that are different colors, and you can actually see that in this camera. It takes a bit of work to pull it together, and it's a lot of logistical coordination, but it's an interesting picture without having to collect the sample yourself. You can have an image, but you still need the sample to have the animals. So it's Still a requirement.
Yeah, what about back here? What are your guys' tricks, tips and tricks for living on a boat?
I'm still figuring it out. Don't look at me. I'm doing the best I can to sleep at night and then just
That's right.
keep
Getting
from rolling.
rest.
Especially
Yeah.
that first night when we were on transit.
Oh,
Oh yeah. Getting your rest and eating.
True, and the sea legs help.
Yeah. I
Just put you to sleep.
let's see, I bring a little stuffed animal that my daughters let me borrow.
Oh,
This time actually she bought me one when she was in Japan this summer, so I have that little critter. And then, yeah, I bring my own pillowcase. Just, you know,
yeah.
it's nice to have
I
something
have
familiar.
my own pillowcase.
Yeah. One of my friends on a cruise that she was on, expedition she was on, she had this little organizer that sits right next to her rack, her bed. And it's nice, you can put everything like the charger and your book and your water bottle, all these things together so they're not going to fall out, fly all over the room if the sea state gets bad. So it's like a little cubby right next to your bed. And so I got a little one that I'm trying out on this cruise. So
That's
yeah,
epic.
it's fun. Yeah.
Yeah, the water bottles really do fall
They go everywhere.
constantly.
Yeah, and they make a big sound. Wake up your roommate.
So I had my water bottle wedged underneath my pillow.
Oh, there you go.
Yep. Yeah, I actually didn't know I'd be joining the cruise until a week beforehand.
Oh,
so
no,
I
you
didn't
have
come
much time to
prepared
prepare.
with all the layers.
Yeah.
So luckily I had a good friend on Raro lend me a puffer coat. It's good to bring layers for this room.
Yeah, really.
In particular, where the AC is blasting. And also the cabins themselves are pretty cold.
Yeah, I've definitely had to put on every layer sometimes.
Yeah.
Our cabin was super cold when we first moved in. I'd rather be cold than hot, but still, it's hot. It can be really cold.
You can always go out on deck to warm up, though.
It's true. Yeah, it's true.
Last night it was raining.
Very hard.
It's a little wet out. Sometimes it's kind of damp out there too, but. Somebody said, what does a typical day look like for people there? Is there an 8-hour on, 8-hour personal time, 8-hour on, 8-hour sleep? So like a split 8-hour day?
Change the 8 to 4 and you're pretty much spot on though.
Yeah, it's a little bit different for us on the ROV team. We
Yeah,
work
that's
12-hour
true.
shifts. So at the minute we're doing, I think our shift's midnight until noon, and then that's it, we're done. Unless, of course, the ROV's going into the water right around lunchtime or something, then we'll hang around, help the guys get it in on deck or back into the water, whichever. But yeah, basically once that 12 hours is done up here, then that's us finished and chill out, personal time, go to sleep.
You see that spot coming up on the top left? Yeah.
Yep.
Yeah, do y'all, how do y'all feel about having the 12-hour shifts? Those must be long.
It's, for me personally, I know it's not what they tend to do on Nautilus. Even, I think on here, the ROV team tend to do the same thing as you guys. The 4 on, 8 off, whatever it is. But given how things have worked out this trip, especially at the beginning, I'm quite glad that's not what we were doing.
Yeah.
Right.
But for me personally, that's just, since I started in the ROV industry, that's what I've always done, is 12-hour shifts.
Okay. It's pretty standard.
Yeah. All right, Dave. You're good to zoom.
What do we have here?
We got a 2-for-1 special.
Yes, we do. Spiky sea cucumber.
Oh, that's right.
Wow, he's really spiky, really
Do you want me
metal.
to come a wee bit closer, or?
No, this is good. We got a
That's fine.
spiky sponge as well on the left. And that sea cucumber is something that looks like what we've seen, at least on a previous dive. It has a genus Synallectes, but I'll double check that.
Good.
Thanks. I would like to get a better idea on this spiky sponge, but
There we are.
to be determined. The spines coming out from it.
What does the
Beautiful.
doubler do to that?
Ooh.
Wow.
Ooh, lovely.
Ooh.
That should be definitely good enough to get
Yep.
in the
Yeah, for sure.
Just
Good,
another
98.
minute, Dave.
Thanks, pilot.
Yeah.
That's great.
I'm looking straight,
On the
almost
move.
straight down.
Yeah. That sounded just like a
Is that
clip out
a
of the
crinoid on
Minions
the right
movie.
too?
Ooh. It's kind of like a like a desert here, because I know in the desert.
There's your fish.
Fish.
Oh,
Ooh,
first
ooh,
fish of the
Is
shift.
is that a
That's
big
all
one?
you had to say, and now the fish
I
appeared.
know. They didn't like that.
Time for the fish show.
Ah, get into the light. Go on.
So I'm definitely seeing a long dorsal and the anal fin that run the length of the fish
Up at
itself.
you.
Yeah.
That's a
Um,
good
Getting
shot.
The
behind
head
me.
has a kind of characteristic
Yeah.
shape,
He'll move.
and I guess it's a type of cusk eel, which is in the Ophidiidae family.
There he is.
There he goes.
Oh, hello.
He's back.
Maybe his face will come around. Show us your
Yeah,
pretty
he's-
eyes.
Decided he doesn't want to get tickled by thrusters after all.
Yeah, it's not a good way to go.
No.
All right, I'm going to herd you up there.
Yeah.
But what I was saying is that in deserts, most of the actual biodiversity is underneath the ground. It's like 99% or something. So
I didn't know that statistic. That's interesting.
it's actually, yeah, there's actually
Oh.
a lot of life in deserts, but you can't see it. And then here, like, there's so many spiky guys too.
Speaking of which,
Kind of like cacti.
Asako just commented the last watch saw one and they recorded it as an urchin.
Oh, really? Interesting.
Oh.
Thank you, Asako.
Thanks, Asako.
It's always good to have someone on both watches.
I'm not sure we have a record of that urchin in our database, so we'll take a look.
Saying roughly 63-ish minutes ago.
Yeah, I was wondering in our photographic database that we've been using the APSMA, if there's other records of these
There you go.
deep sea.
That's about 45 there.
Interesting.
Okay, mate. Thanks.
Yep.
Oh.
I'm not really sure about that ID.
Something to be searching for during the
Yeah.
next break.
We have a lot of homework to do.
Yes, we do. There's
Oh, the cucumber, like Panacea, purple Panacea. Yeah, there's, that's a good point, Lisa. I'll look that up in terms of the holothurian. There are other abyssal plain sea cucumbers that resemble that one. The genus Synallectes. It has a lot of those sort of spiky spines. The
actually.
morphology or the shape and the look of it are similar, but
Amanda,
yes.
about that for similarity.
Yeah, it's just a little, it's more rounded, but
It was more
maybe.
rounded.
Yeah, we'll take a look at it. Thanks. Thanks for the fast pull. That was awesome.
I flipped to the right page at the right time.
Oh, you ended up going with the book, huh? Not with the laptop?
I forgot the laptop downstairs.
Oh, well.
It is officially way too far away.
Yeah, I'm definitely open to alternative IDs out there. There's a lot that I am still becoming familiar with in these abyssal environments in terms of taxa. So
Hey,
I
Nav,
love all of the feedback.
Nav, can you scroll over on HiPAC? Where are we going next? Is it the same? Hey, Nav. Hey, Nav. Can you scroll HiPAC over? Is waypoint 6 in the same exact direction, or are we making a turn?
Yeah, it's in a line. I can show you if you'd like.
Yeah. Okay.
Yeah, there's waypoint 6. Can you see it there?
Yep, thanks. Are they already dialed in to keep that steady move going, or?
I readjust them every single time we hit a waypoint.
Yeah, okay.
So, I mean, if you want, I can keep out, but I should probably zoom in so they can track.
Nope, I'm just curious. Thanks.
Other
Appreciate
alternative.
No worries.
What's our layback right now? We're way back there, right?
Yeah, it was about 300 meters earlier. Let's.
300.
Yeah.
Okay. Wow.
Yep.
Yeah. If you stop the ship, please holler.
Will do. Yeah, not planning on it.
We'll come swinging down if we stop.
Okay.
Thanks.
Oh yeah, sure. Can we take a look at this over here on the left?
Okay, Dave. Thinking another part of a shell, maybe?
Is that a tooth?
Kind of looks like a tooth.
That's
Yeah,
interesting. It does look like a tooth.
Yeah. We did see a lot of shark teeth on the last watch.
Yeah.
Most of them were old and coated black colored.
A brand new nodule. Yeah, that's a tooth.
Yep.
Thanks, pilot. Appreciate it.
Yep, on the way. Thank you, Davey.
So yeah, I think our shoreside scientists think that small, round, white, spiny animal is more likely an urchin. They noticed some tube feet were moving, and let's see, Mike was able to look up to the database, and there might be another one that's been identified from the abyssal plain, different region, but still our best resource at this point, along with our shoreside scientists for information. So we'll have to add urchins to our list of taxa that we've encountered so far on this dive. That's exciting. Another type of echinoderm.
And I found another image that might actually be even closer.
Oh, good.
Although I think that one still has too many spikes for what
Oh,
we
yeah,
saw.
it looks
Similar.
super spiky ping pong ball. Wouldn't want to touch it.
Someone said pteropod shell.
It could be a pteropod test. It looked more dense and tooth-like, but that's a good point. We've been seeing the pteropod tests on the seafloor earlier on the dive, but had more of a characteristic tooth shape.
We should, me trying to read all of these names is like kind of funny because they're so hard to read. We should make a series of going around and trying to have people pronounce some of these organisms.
That's good.
The pronunciation
You're
bee
doing great.
instead of the
You're doing
spelling
awesome.
bee.
There are all these new words.
So if two organisms are sitting right next to each other, does it mean they're best friends?
Either that or food. It's one of the two.
Keep your enemies closer.
Yeah, next time we see another white ball, we'll have to take a closer look at it.
See if we can spot tube feet.
Yeah, I think we'll be on the lookout for those tube feet. That'd be good. That'll be good for our records keeping as well. Looks like another shark's tooth in the bottom right. So we started at waypoint 4 on our dive?
We started about a quarter of the way past waypoint 4.
Oh, okay. Starting to feel like I'm achieving something, but we got a little ways to go. We're halfway there, everybody. The dive plan today is to essentially run a line, a really long 10-kilometer track through 10 waypoints. Each of them are about 1 kilometer apart. And image as we go, collecting data and annotating what we can find along the way. The terrain has been relatively similar, definitely not as variable as our first bit long dive, and primarily composed of a variety of several rather high density manganese nodules with a fine sediment coating or powder. The organisms that we've been finding have included a number of different kinds of sponges and echinoderms, including a few different kinds of holothurians or sea cucumbers, crinoids, as well as looks like sea urchins. And we've been seeing a few fish so far, and on the last watch they saw black coral. So not a comprehensive list, but a lot of diversity in such a small distance. And we're really only basically carving out our observations on a really small section of seafloor within this broader Cook Islands region. So it's a it's the tip of the iceberg, so to speak, just the beginning in terms of us understanding what's living down here.
Makes you really just want to set something tasty down there just so you can wait and see what comes to it.
Yeah. Baited traps. Yeah. I know John was talking about some other baited camera systems that they had deployed, and they observed a variety of different scavenger type species that responded. And it took a little time for them to come in and sort of, I guess, smell it. And several different fish species came in and different kinds of crustaceans. So again, it provides us an understanding of a different kind of community that we don't normally encounter or don't usually get to see on these dives. Sometimes they're few and far between and scattered and spread out.
Astrid from the Science Portal is asking which fish we're seen so far. I think mostly they've been cusk eels, but if I remember correctly, they mentioned that there was a juvenile that was seen in the last group, but they couldn't quite figure out what it was.
Yeah, thanks Astrid for tuning in. That's about it. I believe we got some good images. I hope we'll be able to further ID what they are. Yeah, I'm curious about the other fish groups. The tripod fish species, we haven't seen any of those. We saw a lizardfish, but that was on the previous dive in a different area. As far as the Typhlonis, the blind fish, I think that we may have seen that early on at the beginning of this dive, but we'll have to take a better look at the imagery. We didn't get the best face-on view of it, so to be determined. But lots of great imagery to dig through. Thanks for joining, Astrid. I hope we find some tripod fish. They're super neat.
That would be really cool. Guess I've got another brittle star on the bottom.
Yeah. I think it's a Brisingid, so asteroid. Yeah. Not a brittle star. Yeah, good. And then carnivorous sponge on the right, maybe? Can we look at this possibly, pilot? Thanks. Now I want to see more spiny spheres. Just when I want to see more of them, I don't see any of them.
Oop, sorry, good to go, Dave.
We've got 2 Daves in the front row.
Yeah.
Nope. Looks like another sea lily. Crinoid. It's so delicate looking.
Mm-hmm.
Do you want me to come up on the head on that, Dave, or are you good?
Yeah, whatever you like.
If you can a little bit, Dave, that'd be great. See if there's anything else attached. But do it what's possible.
Okay. You can go tight if you want. There you go.
Wow.
Oh, it's beautiful. So delicate.
Some of these ones have these like tendrils coming off the tips.
Mm-hmm.
All right, we're moving on.
Thanks, Dave. Fantastic. Thanks, front row.
No worries. Cheers, Dave.
I think I've seen at least 3 different, significantly different morphologies in the stalked feather stars.
Yeah.
There's this one that has kind of like the light tendrils coming off the tip. There was a yellowish green one that had more of a random branching in the arms. And then a 10-armed one that was a little bit more regular, but with a more classic feather star shape to it.
Bridge is calling you, mate.
Sea lilies are always a fun find.
Oh, thanks, Aaron. Yeah, those are great observations.
Looks like another glass sponge coming up too.
Yep.
I was hoping for one of our spiky
Round
balls.
ball.
That's a stalked
glass
sponge, yeah.
Yep.
There's another left or right, take your pick.
Let's go this one.
No,
Yeah, no worries.
could be.
It's interesting because
Let's
some
take
of
a
them
look.
are so delicate looking and some have such large spikes.
Now there's a reason for
Coming out of
that.
them.
So you kind of have to adjust your main audio, your main level, and then adjust individual
So then
audios.
pilot, there's this one,
For
and
everything
then
else.
in the distance there's something to take a look at if we
Yep.
have time. Okay.
We'll have to be quick. Okay,
Yep.
Dave,
You
let's
good?
go, mate. Have a look, see what we get.
So we're looking for the tube feet. We got a good angle on it here, potentially.
Let me come around slightly. Hold there a second, mate.
It moved.
There you go.
It moved?
Yeah, it just shifted a little bit.
Ooh.
See? Couldn't tell you if that's a current or...
Camera is absolutely
Right.
vertical.
Yeah, okay. There's no feet on that one.
No eyes either.
Interesting. It does look more urchin-like than sponge-like, so I'm
And now that we're looking at it again,
changing my mind a little bit more.
and definitely
It's
not the one that I
moving.
found on
It
there.
is moving.
It is
a wee
definitely
bit.
moving,
Yeah,
yeah.
you just can't
It's
see.
very slow.
Yeah,
Yeah,
there you go.
Nice. Thanks. That's awesome.
Okay, we gotta go, guys.
Go-kart
Falling
mode.
behind.
Thank you so much for that
That was
stop.
lovely.
Yeah, thank you.
All right,
Gonna have
see what
to.
I can do about this other one up here.
Yeah, on the fly. Awesome.
Crinoid, from the looks of it. See the hold fast.
You're now solidly behind Atalanta.
Yeah, okay, I'll stop then. We're on the move.
You're
Yep.
good. Looks like, yeah, I agree with Aaron. I think it's another feather star crinoid.
It's gonna zip off until we catch up again, guys.
Oh yeah,
Yeah, so I stand corrected, which will happen more than once on this cruise. The urchin. I'm so glad we got a good image of it. And that's definitely going to help us with future annotations, confirmation of that sea urchin is really important. Again, the urchins are another kind of deposit feeder, much like the holothurians or sea cucumbers that we've been seeing. So a number of the animals are optimized to feed on that sediment material that we see in front of us, or they grow long stalks to sit up off the bottom and above the boundary layer so they can access food flying past it through the currents.
Yep.
small coral there it looks like. A few of them here and there. Another one of those urchins perhaps right here.
Looks like another urchin. Oh, I'm okay with we got a good image. That's good. Thanks, though.
So when you pick up a sea urchin, it leaves like a scar on your fingers, right?
If you're delicate, yeah, you can pick them up without poking yourself.
No, but doesn't it leave like a line? I had an experience like that as a kid.
Oh, yeah? Where were you?
I was in Tanzania,
Oh,
in Zanzibar, and I picked up a sea urchin, a black one, with like orange eyes, tiny little eyes. I hope this wasn't a dream. But I remember after picking it up, I like swirled it around, realized it had orange eyes, got really scared, dropped it, and then I had a scar on my finger for a couple of days from it.
Oh, wow.
Something up here on the right.
But no lasting damage.
No.
Okay. That's good.
But I know, yeah, I never asked anyone about it.
Oh. I know their spines can cause some damage. People have been poked in a variety of different places, accidentally stepping on them. Snap zoom if we can, Dave. Ah, it's a sponge.
Oh, wow.
Partially dead and partially alive.
Yeah.
This is a type of glass sponge. We can see the dead tissue on the right-hand side, sort of brown material, and the live is on the left. Thank you. Thanks, pilot.
So somebody asked, isn't it ideal to get ahead of Atalanta, or does each pilot have their own preference?
We are ahead of it 90% of the time until somebody asks us to stop for too long.
Somebody.
Yes. Not,
Who
not
would
sa-
do
not
such a
pointing
thing?
in any fingers. But you can't get too far ahead of it because that yellow flying tether that, I'm not sure if you can see it on the feeds or not, is only 30 meters long, and we need to keep a vertical separation between us as well. So
Oh.
we've really only got about maybe a 10 to 15 meter window to operate in.
This is the first dive for that yellow tether.
Shh. Say things like that.
It's doing great.
It's the best flying tether ever.
Best ever.
Yeah.
Well
I wanna
tested.
upset it.
We're not tempting fate, are we?
Your ROV shift for this watch is rebuilt it 2 days ago from scratch.
Oh, no.
Like, seriously, let's just stop talking
Can
about
we
it,
stop?
please.
It's like, there's
that
the
I'm
question, the
superstitious or
precursor for
anything.
everything.
How about
But it
that?
can hear you.
I've been seeing a few things coming up out of the sea, the bed here, but kind of between the nodules sometimes, that are more like a brownish mushroom, almost like looking structure. I don't know if
Oh, if you can
We've-
see some, like, mushroom-like object on the nodules, please shout it out, because that would be neat to zoom in on.
would those maybe be like the xenophyophores.
They could be.
Yeah.
Yeah, that would be good to get some images of that.
Unfortunately, I usually can't see them until we're right on top of them.
Oh, yeah. I know we've seen small foraminifera, they're single-celled organisms that come in a variety of shapes and sizes and types when we've zoomed in on the nodules themselves.
Mm-hmm.
But what Aaron's describing is a particular type or morphology that would be good to get some close-ups of. They're more like megafauna rather than microfauna or myofauna.
Even though they're still single cellular, right?
Yeah, because they get so big, they create a lot of structure as well. And then with some of the xenophyophores, people have studied what lives in them and then what lives underneath them, and they're really distinct communities of organisms associated with the xenophyophore habitat that's different than the surrounding environment. So a lot of changes can occur in the communities over small spatial scales.
There's
Is that another one of those urchins on the right?
Yeah, now we keep seeing them.
There's
Now they're everywhere.
everywhere. There's 2 of them there.
Yay.
the one on the left might be a stalked sponge.
Yeah.
Yeah. I think I see it casting a bit of a shadow. Yeah, just passed over some of those mushroom-looking things.
Go there. Someone said, thank you for doing this. Super interesting.
Oh, thank you. It's really interesting to me too.
Oh, what's this right here? Dead center towards the bottom.
Where?
You're looking
Color?
at the
I
very
got to
small.
pass over it, yeah.
That white thing just
No,
around it?
no, it's closer than that. It's like right at the bottom of the screen at the moment.
Oh.
I'm going to
Yeah.
have to give Aaron access to the telestrator.
No, don't do that. We won't move anywhere.
Aaron is
Little
a,
Okay.
etch-a-sketch.
Aaron's a fantastic extra pair of eyes. He's been
Yes.
catching things left and right and center.
Yeah, so if you zoom in right there, we should be right on it, basically.
Right, I don't know.
I don't know what we're looking at.
Are you
so.
looking at that dark patch?
It's not that he has better eyesight, it's that he has a better monitor.
No, he
Okay,
doesn't have a
let's
better
see.
monitor.
Could you go ahead and push in, Dave?
Dave? Yes?
Yeah.
Yeah, sorry mate, go for it.
So you see down, like those stalk looking, oh, is it like
Right,
a
got
remnants
it.
of a sponge, but there's like lots of little things on it.
To the left a little
This
bit?
thing?
Or?
Yeah, just to the right of center here.
Oh,
So,
Right, hold that, let me see if I can swing around onto it a
Yeah.
bit better.
oh, good eye.
Wow.
Is it maybe a remnant of one of those, like ping pong sponge?
That's a
Maybe?
good question. It could be the stalk of a sponge and maybe some
Okay,
other
Dave, you can go
animals
tight.
living in on it.
It looks like it's got some critters on there too.
Yeah, it could have those look a lot like tubes of something else,
Have you tilted down already?
but it may be a
I'm
dead
down all
sponge
the
as
way.
well.
With maybe a
You
couple
got a couple
small.
minutes.
There's something going
Isopods.
inside one of the
It
tubes
looks
right
like,
there.
yeah. I know amphipods build tubes like that, so it may be that they've
Okay,
created a
that's
Kind of
little
all right again,
colonized
mate.
stock.
high-rise apartment building on
Gotta
this
go.
stock. Yeah,
Well, thank you. That
thanks. That was good. Neat.
hadn't seen
Yeah, it's
anything
like
quite like that.
you can see that there are some organisms that we're not going to be able to capture unless we're basically running inch by inch over the seafloor looking.
Right.
The little Herc was here.
Yeah,
That's right. That's right.
there's only way I could get low enough to see that.
That was good. Really neat. Thanks, Aaron.
And good spotting, too.
Start tilting up.
Yeah.
Wow. Lots and lots of nodules as far as the eye can see.
Getting some larger ones here and there.
How many trillions was John estimating again?
There's over 700 trillion,
nodules.
plus or minus.
Plus or minus.
Is that individual nodules or is that by mass or something? Or
Probably individual.
wow.
Yeah.
Somebody popped in and asked, if Little Herc is used for visual exploration, what kind of equipment does Big Herc have that makes him different, besides the obvious size difference and different depth limits?
That's a good question.
Well, Big Herc can collect samples, right?
Yep.
That's probably the biggest difference. It has a couple of different sampling techniques on it. The manipulator arms, of course, are able to reach out and collect physical samples, and there's some bio boxes on board that we're able to stow those samples in until Herc reaches the surface. And then there's also a set of push cores, I believe, where we're able to push those into the sediment in order to take samples of the seafloor sedimentation, and that also pulls up organisms and other material with it. I think there's a slurper as well. I'm not sure what the scientific or proper name for that would be, but it's essentially a vacuum that pulls in some water, and then anything it slurps up gets stored in another type of bio box.
I'm pretty sure the scientific term is a slurp gun.
Yeah, I like slurper.
And
I do like slurper.
The other major one would be the Niskin bottles, is that right?
Okay, Dave, let's zoom in on that bit, please. Thank you.
Oh.
Hang on now, come around a little.
Whoa. This looks very crustacean-like.
Fancy crustacean.
Whoa. Very interesting. Its back end, or sort of telson, it's kind of flipping up. So we're looking at a type of crustacean. It looks like an isopod to me. Very different, distinct.
Okay, Dave, come wide
others.
again, mate. We're on the move.
Thank you. Thanks for that.
Yep.
Something different.
Very interesting. Astrid is also thinking isopod.
That's my best guess, Astrid. I think so too.
Lisa says Storthingerinae.
Oh wow, you're getting the good ones. I'll have to look that up, Lisa. Thanks. The A-R or O-R?
Storthingerinae. We should have a spelling bee too.
Oh, that would be fun.
They did do one for the last, for the last dive.
A spelling bee?
Yeah. I'm pretty sure it was Teudu that was on here, and she was going through and doing a bunch of Cook Island words for the second portion of it.
That's epic.
We were also doing some of the trickier different species and phyla, like ctenophore. It's like, C is for ctenophore.
C is for ctenophore. It's like the license plate game.
We're just swapping out seats here, and I'm getting up in our ideal position, and then you guys can start
in the front
pointing
row.
some stuff out soon.
Oh, right on.
You're ready.
Yeah. All right. All right, we're doing pretty good now. So I'll slow down. Might have a little bit of a stalked something up here.
Oh, a stalk something.
Do you want to come down, Dave, a bit? It's all good. Yeah, there we go.
Quick shout out to all my colleagues at Deep Sea watching in the office. It sounds like the day's distraction is up.
Aaron will get back and nothing he'll have gotten done on any project.
Yeah.
That's all good. We've been working very hard on this camera for some time, so I'm glad that everybody's getting a chance to.
Anybody want to look at this?
going. It's
Okay.
another sea lily.
Roger.
Or
Is that what they
crinoid.
are? Sea
Yeah,
lilies?
yeah,
Nice.
type of crinoid.
I haven't heard that term before.
Yeah. Common names are tricky sometimes to fall into, but I like that one. I think it's pretty.
It is pretty.
Well, to the Deep Sea Power and Light folks that might be listening, job well done.
Yeah.
It's a great camera.
Yes, thank you.
For real, it's super fun. We're enjoying playing with it.
We appreciate letting Aaron, or borrowing Aaron for the duration of the cruise. He's been able to pick out a lot for us to see and image. Really incredible.
I'm selfishly taking advantage of the opportunity to be able to stop and get closer pictures on the interesting things, because I usually just yell at my screen at home. It's like, oh, what was that?
You mean they don't hear you through the TV?
No.
Years of pent-up excitement.
Been training for this for a while.
So could you tell us a little bit about the light
Well, there's a sea cucumber.
and the camera system?
Sea cucumber on the
Oh,
left.
yeah, I guess it's been a little while since I've done my camera and lights speech.
For those people just tuning in.
Yeah. So the reason that I'm here on this expedition is that my colleague Mikhail and I are with Deep Sea Power and Light out of San Diego, California. We design and build lighting and imaging systems for deep sea exploration. So currently Little Herc is fitted with our latest UHD subsea imaging system, the MXDC cam, and a suite of our high color quality LED sea lights. And we're essentially conducting a field trial with this in collaboration with OET, providing the system to be able to do this deep water exploration. This is the first 7-kilometer rated version of this camera that we've produced.
I feel like this chair is coming loose. Sorry.
Yeah. And yeah, we're
awesome.
I know, but I think it's coming loose, so.
I'll have to get tighten those bolts down for you between shifts.
Don't need it falling off.
No, for
Yeah,
real.
that's dangerous.
Yeah.
And you said this camera was rated to 7 kilometers?
7 kilometers, yes.
Wow.
Oh, wow.
Let's go.
So I think last shift we talked about how Herc can reach or access about 98% of the seafloor. Our camera at 7,000 meters just notches it up one more percentage point to about 99%.
Yeah,
So
you want to look at that?
That would be great. Thanks, pilot.
And right now this is the deepest dive for this camera?
Deepest dive for this camera and deepest dive for Herc.
Little Herc.
Little Herc, sorry,
Yeah.
Little Herc. It would have been the deepest dive for Herc too. We'd be in trouble.
Yeah, right.
We wouldn't be getting it back.
Okay, go ahead and zoom.
Interesting. Oh.
So we have a whip coral. No,
It's just like a
those
little,
are all
Those
little.
are all little crustaceans. Some amphipods.
Several.
Weighing it down.
No. On a
Wow.
stalk. I don't know if the others are amphipods or isopods.
They're definitely doing a good job mimicking coral. I wonder
Wow,
if that
I know.
pose is a similar
Yeah.
function. They spread out like that in order to catch
Food flying by.
marine snow flying by.
Neat.
all
You
right.
see kind of them
Definitely
hanging on with
an
their...
amphipod is the large one.
Are you guys happy with that zoom?
Yeah, that's lovely. Thanks. Thanks so much, pilot.
You can come wide.
Not sure about what the stalk is, if it's from a crinoid or from a sponge.
Oh, here's one of those greenish yellow
Yeah.
crinoids.
Can't go too far.
Stalk crinoids, yeah. We've seen a few of them. This might only be the 3rd or 4th I've seen since coming on ship.
I'll just fly over this one if that's all right.
That's fine.
Yeah, we've gotten good images of them already.
Yeah, I think I remember us seeing a few of them on the last.
So Amanda, I'm wondering if you can...
What'd you guys say it was? Sorry, before we go too far.
It's this yellow-green crinoid.
Okay. Thanks.
It's a little coral, maybe?
This one.
We have another
or
camera
sponge.
Is this
question.
something you would like to see?
Yeah, if you can, that would be great.
Yeah, we'll just do a quick one.
There's something
go
next to
ahead.
it there too, it looks like.
Tilting you down. And yeah, just get in there as best you can.
This one's going to need the doubler.
And then we got to move
the
on in a
doubler.
few. Yeah, all right, I'm moving on.
Thanks,
You can
pilot.
come wide.
I'm wondering if that's a type of bryozoan.
So bryozoan, what's that?
It's another group of organisms. I feel like they might have seen them earlier on in the dive.
And I'm just going to kind of catch up to Dave here.
It's a whole different phylum. And that morphology is pretty similar to some others that have been seen here. They create different color colonies. They can be white, they can be kind of greenish.
This looks like a we're dolphining pretty hard.
So in that case, we would call it other bry ? I didn't get it when we were flying by, but never too late to annotate.
That was the thing we just passed over?
Yeah.
Yeah.
Lisa says her favorite isopod, other than the giant ones, of course, are the cerulid isopods. They remind me of trilobites.
I'm just getting more center to your
Cool.
thing, Dave.
And Steve online on the science portal was mentioning it could have been either the sponge or a crinoid stalk.
The one with the amphipods on it?
Yeah, with all the amphipods.
Yeah, I think I can't confirm or deny which one it is, what those guys were living on, what those animals were living on.
It's another high rise.
That's right.
So have we found anything that you wouldn't have expected, Amanda?
Expected. I think we've seen more variety of organisms than I had anticipated, given what is known about what we call the productivity regime within this area. It's been generally considered oligotrophic, so low productivity in the surface water column. And so that kind of limits the types of organisms that could live down here and thrive. But we're seeing a lot of variety of what I would consider relatively high diversity. So that's been really eye-opening. And maybe we can say that there's, sufficient amount of food to be able to support the variety of organisms we're encountering, sufficient either in time or over, if it's variable, the animals are still able to make a living. If we have time to take a look at that,
yeah, we
pilot,
do.
that's great.
Also, the ROV just passed waypoint 5. We're halfway there.
Halfway there.
Oh, wow.
Are you guys going to start singing?
Scoot over a little bit. Yeah.
Everyone breaks out in song.
You can fly in a little closer, right?
Yeah, sure. I just try not to get in the dark pool here.
Yeah, understood.
So what determines how long the dives are?
The weather?
Yeah, there we go. Okay.
True.
All right, Dave, I'm stable.
Somebody asked, how do you keep the camera cool?
Seawater.
Yeah, seawater is our cooling system for everything.
Yeah, we actually
Yeah, you
spent
can push in as much as you want, Dave.
quite a bit of time, and this is was mostly done by Mark Chafee at...
It's the little clear structure on it.
Oh, this is
all
really neat.
tissue.
So
Sorry.
cool.
Is this maybe a black coral?
It looks like it could be, Aaron.
There's something else there
zone
I'm not
underneath it.
seeing like where it attaches to the nodule, but
But you see sort of the darker skeletal structure underneath.
yep, you're right.
And then
It
what's
looks
this?
like a black coral. There's you can see basically to
hydroid?
the tentacles coming off of
We
the
got about
coral.
10 more seconds here.
And then the dark axis that Aaron was mentioning that gets the black coral its name. That's the skeleton itself that you can see through that translucent tissue. Lovely.
All right.
Nice shot.
Nice.
Nice shot.
Woohoo!
Somebody's asking if Steve is on the science portal. Steve Oshkovitz?
Yep.
Sorry, I gotta tilt up a bit.
Oh,
I think that's a yes. I think
Steve might have a possible ID
that's
for us
annoying.
already too.
You're up is down. There
I know,
it is.
yeah.
There it is. You got it.
It's also got a zone kind of in the middle that likes to drift a lot, it seems like.
Interesting.
Cool.
Yep.
I'm just trying to note for the record. Yeah, I guess they imaged a black coral earlier. I'm not sure if it was the same genus of black coral. The black corals are Antipatharians, and the genus of this one was Abyssopathes. And confirmation from both Steve Oshkovitz and Asako Matsumoto. It was also collected on the Okeanos Explorer on a cruise at a site near Kiribati. The HOL dive, which is a super deep dive that we did with D2.
Is any of this?
I think we're okay here.
Define
Pilot.
super deep.
I want to see it
What is
was
this?
almost 6,000 meters,
Okay.
but Steve's online to
What is
confirm.
this?
His brain's a little younger than mine. Yeah, it was
Ooh.
5,700 meters.
Is this just a big,
This is
What
another
is
sea
that?
cucumber.
is it 2 of them?
it's really big.
It looks
Or
like
just
there's one. It's got kind of a sail to
Oh
it.
yeah, I see. Cool. Are you alright if I do a flyby or?
Yeah, flyby's good. Thanks.
Yeah. Oh, it's big. He's got like 2 prongs on it.
Yeah.
So for those of you just joining us, we are currently exploring the distribution, diversity, and density of benthic megafauna, substrate types, and environmental conditions. We're approximately 175 nautical miles north of Aitutaki, and we're planned to go to 5,104 meters down. We have people visiting from the United States, United Kingdoms, Canada, Germany, Singapore, Russian Federation, Peru, Mexico, and France.
And the rest of the Cook Islands is only just waking up.
Well, that's exciting. It's always good to see some corals that are familiar to us. Each of our image records provides us a better understanding of how they're distributed around this vast Pacific Ocean. The fact that it's Abyssopathes, I wonder what the depth range of Abyssopathes has been recorded elsewhere and what the deepest depth is. We might let our shoreside scientists weigh in on that. And I'm adding that they also, Steve Oshkovitz commented that it was Abyssopathes lyra, which I missed on my first round. It's a very pretty name, lyra. Black corals are found throughout a wide depth range. There's species that are specific to different regions and different sort of depth strata, if you will. And the different corals that we investigate in the Gulf for some of our research there include mesophotic black corals, Antipathes. We have quite a variety and high diversity of those.
I'll be back. I'm going to go join a interaction.
Okay, good luck, have fun.
Thank you.
Looks like another one of those sea urchins, yeah, on the right.
They are absolutely everywhere now.
The little white
ping pong balls.
Yeah. Is this another one of the ones you were seeing earlier?
Oh.
Just larger?
Can we see what this floating jelly thing is? Yeah, that'd be great.
Please be a jellyfish.
Is it? Do you want to snap in, Dave? You want to kind of frame it up? See if I can float it.
Oh, it looks like some sort of Medusa type.
Ones.
Oh, nice.
Oh, yeah. I'll try to.
Yeah, you're doing great.
Lovely.
It does look like a jelly.
Oh.
All right,
Oh,
beautiful.
Thanks so much.
Motion.
That's great
So
from
hard
a
to
really
focus.
beautiful
Sorry, Dave.
type of jellyfish.
No worries,
We'll
not
take
your
a
fault.
look
It's
at
moving too.
our records.
All right, let's go wide.
We've seen a few different forms of, Medusa forms, jellyfish, but I'll have to verify what this
that was good,
because
right?
Yeah. That's a good point. I think our some of our screen configurations used to be a little bit different, especially with when we fly Hercules. So like I'm more used to looking up there because we have more views. He's pointing out that I tend to look at the upper screens instead of looking at the lower ones.
Which one's easier for you?
Probably looking at the ones right in front of my face.
Oh,
yeah.
wow. The upper ones are just sort of attractive because they're big.
I didn't even notice I was doing that, so maybe that'll make it easier.
Oh.
It won't give you a sore neck either.
Yeah,
right.
no, totally.
Ergonomics, important.
I didn't even notice that. That must be completely habitual for me. Yeah. Yeah, and the upper cameras and stuff. Sorry, we're talking shop on SPL. We don't need to be.
I can only hear you, so it's a fun one-way conversation. Dave's allowed to say something too.
I was just not on SPL.
Yeah. No, it's good. He's giving me tips.
That's good.
It's nice having people who have been doing this for a really long time letting you know.
And now she just said I was old.
I was wondering if you'd catch that.
A few of us are on the older side, Dave. It's okay.
You've been doing it for a really long time.
We're experienced.
I'm in a
really
good position
long time.
again,
That's
so
nice.
yeah, you want to see that?
Yeah, that'd be great.
Let's go see that.
Curious about what that jelly was, because we have a long list of homework
for that?
assignments.
You might have to give me.
Yeah, we do.
Yeah. No. I think we're good. I think I caught my toe in the ground for a second there. That's what happened.
It's a good point, though. The nodule field that we see in front of us is a relatively thin layer. All things considered, the soft sediments are right below it.
All right.
Looks like we might have a sea star of some sort on the left as well.
A sea star.
Let's see if I can. All right, go ahead. You can zoom in. I'm not quite centered up, but get in as much as you can.
Well, I'm seeing amphipods.
Yep.
And
I'm seeing about 5 of them on there.
the structure,
We got about 10 more
Maybe
seconds.
6.
I don't know.
Okay. All right. That's good to know.
Structure maybe bryozoan? Or a type of sponge? Or something else? Oh, that's fine. Wow, I'm stuck. Sorry, folks.
Are you all right if I pick up?
Yeah, that's great. Thanks
Okay.
for the image. That's awesome.
Can come wide, do you?
But lots of associates on that one area.
Okay, I'm gonna try flying by
colony.
the. Whoa. Sorry.
Yeah, the depth is to the point where we just almost have to rely on cameras for
Yeah.
spacing.
Yeah,
so.
yeah. Oh, yeah, that was not me being like.
Oh, yeah, I was like, I looked
Just,
at it too, and I thought the same thing. I was like, that's all over the place for USBL hits.
Well, and when we're with Herc, we're usually solving with the DVL as well, right?
No, yeah, it's absolutely what we use. So
yeah.
this is really far down.
Yeah. Oh, yeah, I see. Yep. There we go.
It's actually a great case for why we have DVL in the first place.
Yeah, I definitely noticed a difference. It's interesting. Yeah. Zoom in a little bit. Okay. Yeah.
Well, that was kind of flexible, so maybe it was kind of hydroid?
Yeah.
Not entirely sure. It was really cool looking, though, that's for certain.
Yeah, for sure. So this one has kind of a fuzzy morphology.
Roughly.
Roughly.
Yeah.
Branching. Well, we'll see.
Was that sponge or coral?
It's one of those, yeah.
Yeah. Okay. Mystery.
We're still,
Yeah.
with
or what's the third thing?
I think that would be closer.
Bryozoans.
Bryozoan.
Yeah.
Okay.
Bryozoan's another phylum altogether. So it's
well,
we're starting from the basics.
Everything is either a sponge or not a sponge. So
there you go.
Which means we're halfway there from the start.
Oh, we got a nice little twofer here if you got time.
Yeah, we can frame that up. Just getting a little closer.
Looks like Steve is leaning bryozoan, which is good.
Give him another meter.
Looks like we've got a sponge on the right. And
just getting us better.
What do we got on the left, Mike? What do you think?
Looks like another a brittle storm, maybe.
All right, gotta count the legs.
Can push in a little bit.
Well, maybe not a brittle star.
Oh, beautiful. Yep, looks like a glass sponge on the right.
And what's on the left?
I think it's the Brisingid sea star.
Yeah, okay.
Brisingid. That's the word I was...
Sounds
beautiful.
Good. All right. You good? Yeah, well, that's why I'm using stick lock more than I usually would to do these.
Is there an advantage to everything being white? Or does it just not matter down here at all?
I think they were talking about that yesterday, weren't they?
Yeah, we were talking about it a little bit yesterday. Keep hearing that a lot of the time it takes a little energy to produce any of the pigmentation. So at this depth, since it's so dark, it doesn't require, and energy is so little to find, that they just minimize energy use and don't produce pigmentation.
I'll go a little faster, so.
Which
I
makes
just want to get
more
like kind of
interesting when
ahead
we
of
do
you,
find
and
the red
then
ones, I
that way
guess.
if we look at something, it's not immediately.
So the sea cucumbers are just an exception.
Well, there are some sea cucumbers that are just plain white as well, but there were a couple that were like a nice pinkish red as well. So, and the shrimp that we saw earlier, that was also a reddish coloration.
Right.
Oh, so it's our light. It's, oh.
Huh.
So our shoreside scientists have chimed in about corals, so we're not talking about the bryozoan anymore, but we've moved on to corals. The black coral that we saw earlier, there may have been up to 3 individual colonies observed so far on the dive of Abyssopathes. And then Asako mentions that we also observed Schizopathes, which is another type of black coral. I have to look back at that. I don't know what that one looks like.
Part of me keeps waiting to see if the landscape is going to change at all, and it's just consistent all the way through.
Yeah, I mean, for the last 6 hours. It's been this.
Yeah, I don't want to spoil it, but it's not going to change. My elevation layer
Let
tells
them
me so.
believe, let them believe.
So saith the navigator. I think you might be right.
I mean, they do control the outcome. If it starts to change, they can just turn around.
Yeah, well that, and we have surveyed the area before with the sonar, so.
Sonar looks a lot similar.
Yeah,
Pretty
yeah,
consistent.
it's really flat out here.
Is that a...
Which is, yeah, what you expect.
Wow, things get out of the light pool so quickly.
Yeah.
Okay.
You'd have to find a wreck or something really cool for that to change.
Challenge accepted. Looks like you have...
Possible cucumber on the left.
Yeah.
Oh yeah, it's made a nice little track for us to follow.
Yeah.
Yep.
Yeah, it looks like maybe a sponge on the right.
Are these all similar kinds of sea cucumbers that you've been seeing?
Yeah, we've seen
Oh yeah, this is another one with the little...
the sort of sail feature.
Yeah.
Yeah.
Huh. Fly
And
over
I think
the
the
sponge.
white sponge is possibly a euplectellid, which is a type of glass sponge, or hexactinellid. Not sure what this is.
Yeah, we can take a look at that.
That would be
Looks
great.
kind of similar to the last one we were just looking at.
I'm just gonna try getting closer to it this time. Find out where my shadow is. Learn. Yeah, that's about as close as you can get, eh? Okay. Go for zoom.
So is today Canada's Thanksgiving?
Oh, I do not know.
Oh. Someone wrote in the chat about that. There's another one on the
I
left
basically celebrate
Yeah.
Halloween, and that's about it, so.
I can't wait for Halloween when we
Yeah.
get back.
Looks like
We-
there's an
we
amphipod
just
hanging
make
out
it.
on there.
Josh
sure
asked
this
what
one's
days
alive.
I wanted off this year, and I said Halloween. That was my stipulation for coming out here. Sorry, Dave, I'm trying to get
Yeah,
past
no
our
worries.
dead zone, but it just drifts. Okay.
No worries.
Yeah, you should be able to push in as far as you want there. Just, yeah.
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 Looks like it might be mostly dead, but it looks a lot like what we saw earlier. Just sort of coated in material.
Have we identified what those little, like, sea ants are?
So I think this is a type of isopod. It's a kind of crustacean, like a little itty bitty crustacean.
Come wide. I'm gonna move on.
It's in a different, totally a different group of animals than like the shrimps that we've been seeing. I don't know if we have time to look at that later.
Do we have a minute? No. Okay. Sorry, I gotta move on.
That's fine. I think it was that one on the left is more bryozoan.
Okay.
Maybe like 2. So
tilts up.
And Asako sent us another message saying the ID was confirmed.
Yeah.
And sent images to Tina Molotsova.
So that's specifically about the Abyssopathes coral that we saw way back?
Yep.
Yeah.
Just gonna go over here.
Yeah, thanks to Asako and Tina for the ID confirmation on the corals. That's awesome.
The seat moving around is...
When I get back to shore, I'm going to be home for about 5 days, then going to Idaho in time for Halloween and go trick-or-treating with my 4-year-old granddaughter.
Oh, that's awesome. Do you know what she's going to dress up as? I don't know what that is.
Do we need a closer look at that?
If you can.
A mix of Disney
really
characters, mostly princesses.
Awesome.
Right.
But it's still undecided.
Might be another tooth or body part.
Here. I gotta come back a little bit. Sorry. Okay.
There you go.
It is kind of looking like a tooth.
I might get it with my dust cloud, but you can push in, Dave, if you're... Hopefully our cloud just kind of passes
Yeah, it just went by fast. Looks kind of tooth-shaped.
Yeah, kind of like the one we found from earlier, yeah?
Yeah.
All right. I'm going to go move on if that's all right.
Yeah, that's fine. Yeah, that's good.
Okay.
Nice shot of the nodule field as well.
Come wide. Okay, I'm going to push out in front of Dave a bit, and then that'll give us some more time to look at the next things.
Is that another glass sponge we're passing over to?
Yeah. Another glass
Just
sponge.
watch. I'm right under you, Dave, so. Yeah, I see. There we go. Yeah. Am I? Oh, yeah, you got me. You got me. Sorry. I'm retraining myself. It's actually quite hard to retrain yourself once you've gotten used to like piloting in a very specific way.
Yeah, I believe it.
Similar with the arm operation, like we have 2 settings for how you can hold the arm, and as soon as you get used to one, you're doomed. All right, getting more out. I'm gonna be looking up, I think. Oh, other way. Beautiful.
Yes.
All right.
Oh, we might as well take a look at this.
Yeah, we're gonna go look at that.
That'll be great.
It's in the right direction. Is this gonna be a crinoid or a Brisingid?
I'm leaning towards Brisingid.
Yeah. How many legs do we got here? So 6. Yeah, it's a Brisingid. Do we... I don't have the ROV data on my screen here. What's our altitude when we're generally flying? Around between 1 and 2 meters?
Yeah, it's usually less than 1 right now.
Okay.
You can push in.
Awesome. And the lasers are 10 centimeters apart?
Yeah.
And we're getting a closer look at this Brisingid sea star. It's a type of asteroid. And one other previous collection of, you can see that's neat.
Wow.
Of a Brisingid in the Cook Islands. There's been 1 megafauna collected from the abyssal plain, and that was of a Fraella Brisingid sea star. I identified it.
Okay, we're good here.
Yeah, let's go wide.
Lovely, lovely. Thank you so much.
So if you bring something like that up in like a sample box, would it look a lot different without all the pressure being applied?
Yeah, it kind of depends on the animal. So some of the corals that we bring up can be like full of water at depth, and they kind of get a little deflated in the bio box. If you can have the bio box well insulated, so it's holding temperature really well, the animals, many of them have done well in the cold, like in a cold van on the lab. It can be really tricky keeping them alive, but yeah, again, the simple answer is it depends on the animal. The urchins hold their form. The animals that are more full of fluid kind of deflate a little. And the corals kind of are hit or miss depending on what they are. But in many cases, they'll still be alive when they reach the deck. And then if you can keep them cold, they can persist in the lab on the ship.
Yeah, interesting.
Is that a, what's this?
Is this here?
Yeah. You want
Might
to
be
see
another
it?
shark's tooth. We can take a look.
All right, push in.
I've been excited to see the shark's teeth myself. But
It's been
Or
quite
are
a
we
few on
just
this
looking
dive.
at
Yeah.
something on the edge here?
Oh.
Kind of just looks like
yeah, I think
rock.
it's a like tooth.
Oh, okay.
Just our frame of reference or perspective is a little different.
Oh, okay, yeah, I see.
But it's coated with that iron manganese oxide coating. Neat.
All right.
Thanks, pilot.
Come on.
Teeth aplenty. And then John said that he brought a couple or at least one on board so we can do a
Oh, he
little
did?
show and tell later. Yeah,
Even better.
I know,
Look at John always being prepared.
for sure. Yeah. So we've seen a few different types of carnivorous sponges and glass sponges on the dive so far. High diversity of sponges, I'd say. Corals, and we saw a siphonophore. So in terms of cnidarians, we've seen hydrozoans and a few coral species and the siphonophore. For chordata, we've seen a couple different cuskeel groups and a tunicate.
Speaking of which, one coming up.
Oh, I just have to do the list and then animals jump out at us.
What are we seeing?
Fish.
Well, add Dumbo octopus to the list, because I would really love to see one.
Thought
of
it was a
those.
fish.
Oh.
Oh,
It's not.
it's something else.
Yeah, we're in a good spot, so let me know if you want to see any of
Yes,
the things
please.
that are coming up here. Want to see? There's also
I think
a
I want
large.
to see this.
I
yeah,
agree.
floating in the water column.
Yeah,
Yeah,
before it goes away.
yeah. Let's go find him. You can see him on Atalanta's cam. I think I'm gonna be
Nice.
coming up a little bit, Dave. Try to.
Hopefully Astrid is watching. She can check out the fish with
Ah,
us.
here we go.
Tell us what it might be.
And I'm gonna have to learn where my light pool is here.
You're there.
Yeah. Yeah.
Oh, looks like a cuskeel to me.
Yep.
Oh, sorry, overshot here. All right, let's go
Sometimes
back up to him.
cuskels or sometimes these fish have associated parasites, so it's great to give us a chance to take a close-up look at the
pushing
face.
in, Dave, if you want to try
And then
to.
along the body, not only for identifying the fish, but also any kind of associates.
And I'm gonna get
Look
my
at
head
that
back over there.
face. And definitely got eye spots. Not seeing any kind of attached animals, so don't think we see parasites, at least on this side. on the outside.
Is it speckled or is
Ooh.
it just the...
And Astrid has given us an
Keep an
ID.
eye on me, Dave. I'm not really
I
paying
love
attention to how I'm oriented. Okay.
Bassozedus.
Yeah, Brisingid.
Yeah.
The ghost morphotype. I love it.
All right. Come wide.
Thanks, front row. Thanks,
Yeah.
Astrid. Appreciate the ID.
Roger.
Cool.
Yeah,
I wonder if the Brisingid comes in different colors. That's why this one's called the ghost morphotype. It's because it's pale. That's why they call it that. Great shot. So often we can't get the fish because they dart away before we get a chance to hone in on them. That was awesome. I think we are still seeing a fair number of stalked sponges.
That is exactly what I was just looking at too.
Astrid mentions that it's uncertain about whether the lighter colored ones are the same species, but there's a Brisingid nasus or another species that could be. I believe Astrid works at Oregon State University. She's a professor out there.
Go Beavs.
It's a good school. It's great for oceanography. I went to University of Washington, so.
Go Huskies. I'm okay with the bees. They have a great oceanography program. Up in Newport, Oregon, about an hour north of where I live.
Oh, is this one of the fun ones that I like?
It might be.
Oh, it is. I love these. I only met them yesterday.
You only met them yesterday?
Yeah.
What do you remember about them?
Something ping pong ball.
Ooh, good job.
He's called George.
George.
You can push in.
Oh, nice call, pilot. It's
Yeah.
a ping pong sponge type of
Push up
carnivorous
a little bit. Yeah,
sponge.
okay. Oh, there, centered. Nice.
I love these paddles.
See if I can pop my
They're
heading over.
They're actually three-dimensional and spherical or elliptical in
Oh.
shape, and it's got. What do you see,
Holothuria
pilot?
on it?
Yes.
I'm learning words.
Oh
big
my
words.
gosh. Yeah. So it has a sea cucumber hanging out. I'm not sure if it's trapped it or if the holothurian's just sort of taken a little vacation here.
Yeah, it's upside down though.
Yeah.
It's kind of an odd angle for something.
I
that's
think it
just
might have
hanging
gotten
out.
stuck. And then there's a couple of brittle stars. You can see the white arms entangled around the stalk of the sponge. Really amazing imagery here. Great close-up.
Oh.
You can see the spines along the stalk and the kind of paddle ping pong shape or lollipop shape.
Are they actually like spherical or are they more flat?
Yeah, they look three-dimensional and kind of, yeah, spherical to me. And then looks like the one like on the bottom is more deflated,
Yeah.
the lower left.
I'm gonna move
That's quite a
on
big,
if we're all right.
yeah, we're good. That's quite a big meal for this little sponge
wide.
guy.
Nice.
Oh, it looks like a
Get
little,
out of
maybe polychaete on the back too. Wow, quite a habitat there.
Sorry, Dave, the way that I leave a
Our
shot,
science
do you
chat's
want me
been
to do
going
anything
off.
differently, or are you?
Nah, you're fine.
Okay. I don't want to give anybody vertigo, but...
Ah.
So that's a very happy sponge.
I would say it's got probably a few years worth of food right there. So chondrocladia is the type of ping pong sponge, type of carnivorous sponge. And where it's been documented before, it can be found on nodules like it was, or resting attached to sediments. That was quite a number of different animals associated.
They remind me of silver dollar plants.
Oh, yeah.
Which I really like.
That's a good analogy, yeah. Oh, Astrid, thanks so much for the additional images for the fish that we were seeing. Also from abyssal depths. And it'll be interesting to have Astrid take a closer look at some of the other images that we've seen of fish throughout the expedition. We might have been seeing a variety of different types of Ophidiids or cusk eels. She can help resolve that, and then also kind of maybe if there's some juvenile forms, you know, some more immature fishes or more adult. But we may be encountering a range of ages or life histories of those fish. I was just reading back through the chat and really appreciate all the additional images from other places of the fish, and then some comments regarding the sea cucumber, confirming the chondrocladia sponge.
Yeah.
And maybe that sea cucumber is a little lunch for a while. Yeah.
Am I just getting pulled on? I think so.
All those additional photos were great to look at in comparison
Here,
too.
let's take a look at this cucumber for now.
Okay.
I think Dave's adjusting some things though.
That's fine.
Video Dave, not ROV Dave.
Oh, okay.
But I am also just waiting for my... Do you want to push in, Dave, when you're... Just look at this while we catch up. See its gut.
Yeah.
Yeah.
Taking a closer look at the sea cucumber. It's not really doing much.
Nope.
But you can see its coiled gut full of sediments on the inside.
All right, we're caught up a bit. I can move on.
Thanks. Thanks, pilot.
It's more interesting than just sitting, waiting.
Oh, okay, good. That was a good idea. And it's great to get close-ups of the nodules themselves as well.
Yeah.
They're little mini habitats. Each nodule is a home for a lot of different organisms.
And got another stalked sponge on the right.
Yeah.
Oh, sorry, did I miss it?
No, you're
Nope,
good.
You're
We're
good.
just shouting
Just
things
commenting.
out. I'll
good.
try to point you to things we want to take a look at.
I'll try to do flybys, whatever we don't, but...
That sounds good.
And a possible cusk eel swimming off to the upper right.
Yeah, it's up in the...
Oh, I missed it.
There's also something up ahead that's bigger. Maybe another... For some reason, no reason at all, the French version of SpongeBob just popped in my head.
Are you gonna
Bob
say it out loud?
Leponge. Yeah, let's go look at that.
It looks like a sponge to me.
Bob Leponge.
Bob Leponge.
It's a fairly good sized sponge if it is.
I want to say that we saw these at the beginning of the dive, our first watch, yes?
Yes.
It was sort of platey and
you got
morphology.
A little coral in the back too. I'm just gonna bring my heading around a little bit.
Looks like
you can push in. Oh,
another type of glass sponge. It almost looks shiny to me from a distance, but we'll get a closer look and then you can see the pores in this sponge, the tiny holes.
Yeah. I really wish the seat was not moving. Sorry. I'm talking out loud.
Is this similar to the sponge we found at the beginning of our dive?
Yeah, Jacob, I think that's right. And then I think off in the distance there's something. I think
if I tilt up, do you want to see this little?
that'd be great. I think it might be.
We got the time, so.
Take a look. It looks a lot like that, what we think is a bryozoan that we saw earlier, and then it looks like there's something else attached to it.
And that tilt hates the exact spot we want to be in. Boop, boop. Sorry, you're getting all the sound effects.
I like that.
This is all the things you don't hear when I'm sitting in the other seat. It's got a little, is that a little tiny crinoid thing?
I think so, yeah, I think that's right.
Alright, yeah.
Ooh. Oh, it looks like there is some live tissue on this one. And then, yeah, it looks like a little tiny crinoid is attached to this, what appears to be a bryozoan. Again, it's a different phylum, the bryozoan as how we classify them here on our annotation. A totally different group. And then there's another organism, crinoid, attached.
Come on.
Thank you.
We're gonna move on.
That's great.
Yeah.
Did you used to watch SpongeBob in French? Were you
no,
learning
I
French?
did do 4 years of French immersion. Don't ask me to say anything except for SpongeBob, but
it's funny the things you remember.
Yeah. I did actually, one of the only things that I downloaded to bring out here was the first 2 seasons of SpongeBob. So if anybody wants to watch SpongeBob with me at 5 a.m., hit me up.
In French?
No, in English.
Oh, good.
Yeah.
Okay. Yeah.
Oh, that would be fun.
Is that another tooth that we're flying over?
Is that?
Looks like it. Or
okay, I'm going to keep going because we're behind.
Yeah, you're good. You're good.
Yeah, don't worry about it.
Yeah. Yeah, that way we get... What was it? Okay. Yeah, let's make sure we're good first. Did you see it in your back camera? Okay. Yeah, let's make sure we don't have a little loop or anything. Yeah. All right. You happy, dude? Okay.
Is that another cucumber you just flew over?
Yeah, I think so.
Okay.
All right, we're back in a good place here.
Okie doke.
I do think this new one feels like it sits a bit different than the other one. Yeah, it's got a little bit of a different memory. Maybe. Yeah. Yeah. Is that another tooth? Or is that just a...
Yeah, I think so.
Okay. Are we sick of looking at teeth, or?
No, I think we're good. I mean, we don't need to look at
Okay,
anymore.
perfect.
I'm not sick of them, but we don't need to look at them.
catch myself looking at that top screen. All right. Locking in.